Well, good morning, everybody. I am really delighted to be with you this morning and to be hosting today's PGC Worldwide Lab Meeting. The title of today's lab meeting is the PsycheMERGE Network, Advancing Precision Psychiatry Through Genomic Approaches to Electronic Health Records. And just a little bit of logistics. So today's talks will be given back to back, and then we will hold questions until the very end. We plan to just allow everybody to be unmuted unless at the end for questions, unless you have extensive background noise and you would prefer to mute yourself. So we absolutely would appreciate that. But we're hoping for some robust discussion at the end. So just hold your questions until then, and then everybody will have a chance to use their voice. So like I said, I'm really super happy to be here with you this morning and kicking off today's Worldwide Lab Meeting by introducing the network. So a roadmap for today's session. Our goal really is to introduce the initiative to you and to share some of our flagship projects, including some of the unpublished work that we're really excited about. And after going through some of the basics, the who, what, where, and why of the network, I'll introduce our other two speakers for today. So we have two talks that really get to the science of PsycheMERGE. The first is focused on principles of phenotyping and implementation in the EHR, and provide some examples of different ways in which this has been accomplished, some of the challenges, and what we're using for different study designs. And then the last talk will introduce you to some of the methods that we've been developing in the EHR and their application to depression genetics and immune biomarkers. And so even though it's the last question in my list, I'm actually going to start off with the why of PsycheMERGE. So there are actually lots of reasons that PsycheMERGE formed when it did and how it did. And so I wanted to go through a couple of the advantages that conducting psychiatric genetics research in the EHR has. So first of all, research in the EHR can be incredibly helpful in just the primary goals of identifying variation that contributes to mental health phenotypes. So this is a really powerful figure from a well-known paper by Alicia Martin and colleagues that was published last year, illustrating the growing problem of poor representation in genome-wide association studies across different ancestries. And so the use of electronic health records and linked biobanks, I think, helps to reduce some of the barriers to participation in research that has contributed to these kinds of disparities. And it improves the representation among those who have been historically left out of genomic studies. So this includes individuals of diverse ancestry backgrounds, but also people with comorbidities, children, and those with fewer resources to seek out research opportunities. But while EHRs help this problem, because the settings are naturalistically more diverse, it's not a panacea. And part of the problem in the US, at least, is that access to healthcare is not universal. And many socioeconomic factors like employment status also keep people out of healthcare systems. So it's important to keep in mind that while this is a benefit, I think, of doing research in the psychiatric genetics in the EHR, it's not a cure for the problem. We also have the opportunity to look at health data across the lifespan. And this is, again, a unique opportunity for kind of the naturalistic collection of data across the lifespan to inform the kinds of questions that we can ask in the EHR. So we have the ability to observe changes over, not only over the lifespan, but also over time. And again, one of the challenges of of working in the EHR is that while this gives us a unique view, we also have to recognize the fact that many of these systems are not closed systems. So people can receive their healthcare out in the community and then come to an academic medical center with a biobank and EHR, and so that care then ends up being separated from their medical record. And so again, while it's an improvement over what we can typically collect in research-based ascertainments, it's not a panacea. So probably one of the most utilized features of the EHR so far is the fact that data is collected across the medical phenome. And so this also provides a very unique opportunity to study relationships between traits that have historically been studied in isolation. And so for our research, this also helps to address the kind of historical separations between psychiatry and the rest of medicine. However, because of differences in the way that care can be delivered, again, it can be difficult to obtain complete histories of mental health care. So again, a great advantage of using the EHR to be able to look across the phenome, but recognizing that there are limitations in data missingness. So another thing that we have been working on a lot in my group is studying the excuse me, the use and value of lab data. And so it's the case that having access to the labs, to the procedural codes, and to the diagnoses also really provides us a window into the ways that that medicine and psychiatry are actually practiced in the community. And so most EHRs now have about 30 years worth of data, which again, not only allows us to look across the lifespan, but also can provide insight into excuse me, secular trends of clinical practice, and provide an opportunity to look at the changes in outcomes over time. And I think this is actually going to be some of the most interesting research to come out of the EHR over the next few years. So finally, the EHR also provides us with space to test out the translation of genomic medicine and integration into prediction models before moving them into the clinic. So it's sort of a translational sandbox to see how these models work and what factors influence their performance. And the hope is really that by engaging in this work, that we can reduce the unintended consequences of precision medicine as it's implemented in real life. So now shifting from the why to the what, here's an example of the types of data that are available to investigators in working in the EHR. So this is a figure taken from a description of the resources available to us locally at Vanderbilt, but it's very similar types of data across the network. And so what we have is basically access to all of the coded data, medications, labs, diagnostic codes, demographics, and then also the data that is embedded in clinical notes. So notes, forms, family histories. And this is typically harder to access, but there are methods available like natural language processing that we implement to try to extract meaningful information from these records. So who's involved in PsycheMERGE and where are we? We now have over a dozen sites across the US and Canada and over 80 investigators who are part of the network. And so the network, if you're not familiar with the eMERGE network that the acronym stands for Electronic Medical Record and Genomics Network. And it really provided a lot of the initial infrastructure that we leveraged to develop the PsycheMERGE network. These are, partners, academic medical centers with large electronic medical records that are used for research purposes and accompanying biobanks. And so we have monthly calls with topics that range from phenotyping to methods development to updates on new results. And we usually have about 40 in attendance on these calls. So it's a very robust community. And I just wanted to take a minute to also say that, that it's been incredibly exciting and humbling to be a part of this community. The culture of the group is really that we, check our egos at the door, and we appreciate the fact that what we're trying to do is hard, and that we need each other. We need to learn from each other. And I think we've all kind of had the experience, in one way or another, of our own version of this Dunning-Kruger curve. It's a hard name to say. And, like pretty much every other data science approach, the more we learn, the more we understand how little we really know. And so, I think we're in this, it's starting to make sense phase, but really we could not have gotten there if it wasn't for joining forces together and the efforts of the community. And so we're really continuing to realize ways to improve our science, and having a community that is supportive and truly collaborative is absolutely key. So nobody is going to do this alone. And I know that I personally owe a real debt of gratitude to the PGC and the eMERGE network for building that kind of collaborative infrastructure that really facilitated the very rapid coming together of this network. And so we also are really intentionally working to make this an incubator for early career investigators. It's been very satisfying to see that many of our projects so far are led by early career investigators. And we are hopeful that these collaborations that are being nurtured right now will stay very active for the remainder of our careers. So I wanted to just finish up by drawing your attention to the goals of PsycheMERGE, which really can be kind of summarized in three main ideas. The first is to discover specific genetic and environmental factors that lead to psychiatric diagnoses and their sequelae, and then to translate these findings into learning healthcare systems, with ultimately the goal of improving mental health care. I'm absolutely delighted to introduce you to our next two speakers, Dr. Jordan Smoller, who's professor of psychiatry at Harvard and Mass General Hospital. And Jordan and I are co-PIs of this project, and I have to say I could not imagine a better person with whom to share leadership of this effort. So Jordan will talk more about phenotyping in the EHR and about some of the flagship projects of the network. And then he'll hand it over to Ms. Julia Seelock. So I'm really thrilled to introduce you to Julia as well. Julia is an incredibly talented PhD student in my lab at Vanderbilt, and she's going to be sharing her work on lab-WAS methods development and its application to depression genetics. So just to, end with a very big thank you to everybody in the PsycheMERGE network. This is a picture from our first face-to-face gathering and dinner at World Congress last year. So we're sad to not see each other this year, but we'll look forward to getting together again in 2021. And a big thank you to all of our administrators who help the funding sources, the PGC and the eMERGE network. And also just a shout out, we are recruiting across all of the sites really in the network. So if you're interested, feel free to contact me at any point via email, and I'll connect you with the right site for you. So I think, all right, I will stop sharing my screen and hand it over to Jordan. And again, just a reminder in case you arrived a few minutes later, we're going to hold questions until the very end, and then we're going to allow everybody to unmute and have hopefully a robust discussion. All right, Jordan.
Thank you, Leah. Thanks. And thanks for the introduction. And I want to say equally that, leading this effort with you and working with this group has just been absolutely tremendous. And it's such a vibrant community in which we are really all learning from each other. And I know that I hope others will be inspired to get involved. The community of discussions that we have taught me a whole lot. So it's great to be able to talk with you a little bit about this. And what I was going to do was talk a little bit about some of the principles of how you approach phenotyping in the electronic health record and give a few use cases, some of which comes from the work of the PsycheMERGE network to really highlight how this kind of thing can work. And I will just start by reminding everybody that electronic health records are an enormous opportunity, provide an enormous opportunity that we really are just beginning to leverage. These are, vast resources of real-world healthcare data that are longitudinal, that are passively accruing. As we speak and sit here, thousands, millions of data points are being gathered in the course of real-world healthcare. And in the United States, electronic health records have been very widely adopted over the past 20 years, so they're pretty ubiquitous at this point. And so I think, this does provide us in the genomics community with some very interesting opportunities. As Leah mentioned, one of the things that has motivated, I think, the PsycheMERGE network is the opportunity to actually think about what's the end game? How are the discoveries that we are making going to really impact clinical care, or at least inform our approach to clinical care? And being at the site of clinical care in terms of where the data come from, I think really helps that a lot. We also have these tremendous opportunities due to the fact that many EHRs are linked to biobanks and therefore linked to genomic and other kinds of omic data. And this is just a small sampling of those kinds of opportunities from around the world, the United States and Europe, of course, the UK Biobank and the Nordic registers and biobanks, Asian biobanks as well. And we're seeing just a proliferation of these kinds of data. The Million Veteran Program and the All of Us Research Program, of course, are very large-scale individual studies that are increasingly going to bring these kinds of data to us. So I just want to step back and talk about what is in the electronic health record. Because while I mentioned that it's remarkably potentially valuable because of its vast scale, it's also got its real limitations. And that's where some of the art and skill comes in thinking about how do we get valid phenotypes out of these kinds of records, which are largely, again, they're accrued in the course of real-world healthcare, not for research purposes, as we may be used to, and sometimes motivated by billing purposes, where the kinds of rich phenotypic data or systematic documentation of data don't necessarily apply. So typically what we have in the EHR in any kind of health system is a combination of two general types of data. What we consider structured data, which includes things like diagnostic or ICD codes largely, or CPT codes, which are procedure codes, laboratory values, prescriptions of medications. And those are vast, 20, 30,000 such variables. And then the even larger corpus of information and data that are captured in unstructured text. So those include every clinical encounter in the notes that clinicians write, discharge summaries, radiology reports. Translating the structured data into a data format is relatively straightforward. There are lots of details and things about transforming to common data models and so on, and how you extract those data, but that's relatively straightforward. It's taking fields or variables and putting them into a data set. The much trickier thing is how to leverage the unstructured data, but as Lea mentioned, there are now well worked out systems for approaching that, utilizing largely natural language processing, which is a kind of interface between computer science and linguistics, essentially figuring out how to parse and reduce to data elements of the unstructured text. And I've just noted this article on the slide just because some of what I'll talk about is captured there, and if you'd like to read more about it, you might be interested in taking a look. But natural language processing, again, is the notion that you can take individual text from a note, and you see that here, and these kinds of things, these parts of speech can be mapped to concepts that can then be sort of systematically parsed and cataloged against standardized ontologies of how to interpret those concepts and how to reduce them to quantifiable variables. And so we increasingly are using these kinds of approaches because of the amazing amount of research of information that's captured there. So I want to just highlight that there are a few different basic approaches to how we think of phenotyping in the EHR. A very simple approach, if you're looking, for example, whether somebody has a psychiatric diagnosis, is to simply look at the ICD billing codes, that is the diagnostic codes that are available. And one might say, okay, I'm looking for somebody with depression or schizophrenia, let's say. So I'm going to find everybody who has a diagnostic code for schizophrenia. That's fairly simple. It's a query. The problem is that that may not be a very valid definition. And at Partners Healthcare, for example, we've done lots of EHR phenotyping and lots of chart reviews and validating what kinds of positive predictive values, that is, given some phenotypic feature or variable, what's the probability that if you really looked at that patient, that's what you would see. And it's remarkably low for a single ICD billing code. In one chart review that we did, only 16% of the time when you had a single schizophrenia code was that schizophrenia. So that's pretty bad. But as I'll talk about, there might be some circumstances where you still want to use this approach. Another approach which has become incredibly useful and popular is the use of phi codes. And this actually was developed at Vanderbilt where Lea is. And that is to take these thousands and thousands of diagnostic codes and group them into close clusters of related codes, map them to a slightly higher order category, which does a couple things. One is it reduces the search space when you're doing analyses, and it also reduces some degree of misclassification because you're not trying to make these fine distinctions, but you're getting the essence of a condition that you're interested in. And typically, when people use phi codes, they require at least a couple of codes in that category to call somebody a case, let's say. Another approach is to use rule-based algorithms. That is, not simply say whether this has occurred, but to have a sequence of things that you need to have or need not to have to meet a diagnostic criteria. So let's say you need multiple codes, maybe you need a medication for that condition to be present, you need other things, you need to not have mutually exclusive diagnoses, et cetera. And those are relatively easy to use and can be very effective. And then the fourth, more complex, but sometimes more robust, can be model-based algorithms that use statistical or machine learning methods to actually train models or algorithms to a standard of positive predictive value or sensitivity or specificity. And those in turn can use for example, supervised learning methods, if you're using machine learning, where you're essentially giving gold standard labels that have been validated, let's say by chart review, and then training a model to identify the key features that best separate the classification of, let's say, cases and controls. Or unsupervised learning, which is a kind of, finding the structure in data without providing the answers essentially to the model. And we're familiar with both of these methods, I think, in psychiatric genetics. The key point about all of this in this field is validation and independent data to avoid overfitting. Especially with these model-based algorithms, it's incredibly easy to fit your model to your data, but the generalization and validity of that beyond your data set may be limited. So that's a principle that we think highly of. A couple of terms that you'll see as you look at things like phenotyping algorithms or any kind of diagnostic decisions are ones that you're probably familiar with: the sensitivity and specificity. That is, how well does it pick, how well does your case definition, let's say, detect true cases? Specificity, how well among people who don't actually have the condition, how often is yours picking up that they don't have the condition? And then what may be more important in some circumstances is the positive predictive value. That is, given that your algorithm or phenotype definition calls somebody a case, for example, what's the probability that they are in fact a case? You also see a lot of discussion of area under the receiver operating curve, which is a measure of the discriminative accuracy of your case definition or algorithm, for example. And that is, how much more likely is a case to be called by the algorithm? Is a case's score or status higher than a control? How well are they discriminated? An AUC of 0.5 means it's really uninformative. It's a coin flip. 1.0 would be perfect, but doesn't happen. And mostly we're dealing in the 0.7 to 0.9 range and feeling pretty good about those. So let me just give you a couple of examples of how these different kinds of phenotyping approaches may be useful or used. And the first one I'll just say, I mentioned that the ICD codes by themselves are problematic in some ways, and still you have to validate your code definitions. But one circumstance where we increasingly are using this, or where it may be relevant, is if you're trying to predict the first occurrence of something. So what you want to know is the first time that in real life somebody makes a diagnosis, what are the features that predict that? And so you are particularly interested in that first ICD occurrence. And just to give you an example from some work we've been doing in the area of suicide, we've taken that approach of predicting the first suicide attempt or death using some machine learning approaches, in this case, a naive Bayes classifier. And again, you're able to leverage very large amounts of data. In this case, we had 9 million person years. Now, what we did was to review 2,700 or more notes to validate that the codes we were using for the case definition were actually, according to expert clinician review, picking up the thing that we thought they were. And in this case, the AUC for some reason dropped off here. I think it was 0.77. The model detected 45% of all attempted or completed suicide at 90% specificity. And what you, again, want to do, that was a validated model in which we do a training and then a separate holdout validation. We more recently have collaborated with other healthcare systems around the country to validate and see whether, again, how generalizable is this kind of approach? And in training models at all of these sites spanning, millions of patients, again, we see similar results, which is encouraging across 5 different healthcare systems. The graph you're looking at there is just the actual prevalence of cases among different deciles of predicted risk. So there are some circumstances where if you're careful about an individual code and what it represents, it might be useful. For purposes of genetic studies, we're much more likely to be interested in things like the use of Phi codes and to do what is known as Phi-WAS, that is a phenome-wide association study. The concept of a Phi code, again, emerged from work done at Vanderbilt and in the eMERGE network. And again, this is grouping highly related codes into a single category. And what I'm showing you here is a screenshot from the Phi-WAS catalog, where the URL is in the upper right-hand corner. You can take a look at that. So if you put in the phenotype of schizophrenia, excuse me, you'll see that there are a number of highly related codes that kind of get grouped into that. And so taking, you know, tens of thousands of diagnostic codes and grouping them down typically into about 1,600 to 2,000 codes, which really has some major advantages. Again, you want more than one instance of the code to have occurred typically. And this is where we began in the PsycheMERGE network. And this is work that was just a tremendous collaboration among 4 healthcare systems in the network to get us started. That was Vanderbilt, Partners Healthcare, Geisinger Health System, where the PI there is Chris Chabris, who has been working with a team there, and Mount Sinai. And what we were interested in specifically here was, we know from many studies done in the PGC and elsewhere that polygenic risk scores are strongly associated with risk for schizophrenia. That is a schizophrenia polygenic risk score. And the effect sizes that have been reported, for example, in the PGC schizophrenia work, have ranged from, let's say, comparing the top decile to the bottom decile, maybe an 8 to even 20-fold increased risk. How would this play out if we could imagine in real-world healthcare systems? Would we see any association? Does it, given the noise of the kinds of data that I've been talking about? And also, could we do a Phi-WAS? Could we demonstrate that the PRS is associated not just with schizophrenia, perhaps, but with other things? And might there be some new discoveries? And just to reiterate, a Phi-WAS is in some ways the inverse of a GWAS. In the GWAS, you're taking an unbiased look at millions of markers and their relationship to a given phenotype. Here you're looking at a polygenic score or a marker and its relation to a whole phenome of phenotypes. So in this case, we derived the polygenic risk scores from the PGC schizophrenia data. We used a typical clumping procedure, but also used and found to be more powerful a method called PRS-CS, which was developed by Tian Ge and in our lab and with others in the group, and which has demonstrated greater power in a number of scenarios. And here we used Phi codes to define schizophrenia and psychotic illness. Bottom line, very strong statistical association with the polygenic risk score in these real world data. And if you look at the top versus the bottom decile, it was about a 4.5-fold increased risk. You'll see on the right-hand side, and these were analyses led by Amanda Zeutlin, who was a postdoc in the lab at the time. But you can see all the collaborators there who really were essential to this. You can see on the right-hand side that there is variability among healthcare systems, not surprisingly, because these are different systems, they have different documentation standards, but put it all together and you see a really pretty clear signal there. If you look at the top decile versus all others, the signal is obviously less, or that is the odds ratio is less, because you're comparing people not just at the bottom of the distribution, but across the rest of the distribution. But interestingly, that is not a very different odds ratio from what we typically use in all kinds of risk factor studies. So for example, in the Framingham Heart Study, the odds ratios associated with things like smoking or diabetes are actually less than that. So this clearly suggests that this is not ready for clinical implementation to stratify people in a very robust way, perhaps. But as a risk factor in clinical settings, it's performing comparably to what we see elsewhere. I mentioned that the other thing that we were very interested in doing was the PheWAS, and these were the results here. So on the bottom, on the left-hand side, you're looking at the phi code categories, essentially, and then you've got, just like a regular Manhattan plot, the minus log p-value on the y-axis, and you can see that across a whole range of psychiatric disorders, we pick up signal in association with the schizophrenia polygenic risk score. But one of the interesting things about a PheWAS is the unexpected things that you pick up. And so you can see associations with urinary kinds of conditions, dysuria and other disorders of the urinary system, synovitis and tenosynovitis. There was actually an inverse relationship with morbid obesity and to a lesser extent diabetes, interestingly. And on the right side, not looking at it from the statistical significance point of view per se, but the odds ratios, what you see is that clearly the schizophrenia psychosis phenotypes are most strongly associated, but a range of others are also associated to lower degrees. So in a very preliminary analysis, we began to ask the question, which is now the subject of a whole work group in PsycheMERGE, looking at what if you put these things together? And so in a relatively simple way, we use this naive Bayes classifier based on the EHR to generate a risk score that was an EHR risk score, and then added in a PRS. And the fascinating thing here, again, very preliminary, and we're doing, I think, led by Doug Rudifer and Colin Walsh and others, Carmel Choi and others, more sophisticated analyses of this question. But in this analysis, first of all, the EHR had a superb area under the curve and a very large odds ratio with respect to risk for schizophrenia in an independent sample that essentially dwarfed the polygenic risk score. And in fact, adding that in was less effective than not adding it in. But there are a lot of nuances here. For example, this is people with different degrees of EHR data, some of them with years and years of EHR data. And so it might not be surprising that adding in a single additional feature, even if it is a genetic risk score, is not actually adding much new information. So intriguing, but lots more to do there. The final thing I'll just mention, because it's very relevant to a lot of the work that I think many people are doing in biobank research, is the use of these kind of machine learning and statistical algorithms. One of the things that we use at Partners or Mass General Brigham, and we're using in PsycheMERGE in many circumstances, is a version of this method that was developed again at Partners, which is called PheCap. And essentially, without going into all the detail, because I know we're short on time, but I would recommend you take a look at the paper if you're interested. The overall structure is we take a data mart of all of the potentially relevant cases, including structured and narrative data. We develop a very broad net of potential features that might go into the classification, both from coded features and also from natural language processing. And there's a pipeline that we use that semi-automates the extraction of the natural language processing expressions and then maps them to concepts and ontologies, which again allow us to create a very broad feature set. And then there's typically an instance of chart review in which we review maybe a couple of hundred charts. 100 are used for training. So these are the gold standard labels that the model must now learn to predict. And then 100 are set aside for validation. We do an internal validation of the training model and then set a diagnostic threshold based on what we would like to see for, let's say, a positive predictive value. We get our final phenotype algorithm. We validate it in a set of independent charts. And at the end, and this is not necessarily a trivial process, which is why it's not something that can be done, very quickly, but we get a validated set of diagnoses. And here's, for example, in the Partners Biobank, a small list for just the first 100,000 patients of these curated or validated phenotypes across a whole range of things. We've done more in psychiatry now, and the positive predictive values are at least 90%. So that's very encouraging. With respect to psychiatric disorders, I think, there was especially initially some skepticism about how good could we do. And just to give you an example from the International Cohort Collection for Bipolar Disorder, or ICCBD, where we asked the question, what about rule-based algorithms versus natural language processing, sort of fancy algorithms? In the interest of time, I'm not going to go into great detail here, but we built and tested a whole range of algorithms, including one based on natural language processing, 3 case definitions based on rules, and one based on rules for identifying controls. And this was a lasso model, and these were the features in the natural language processing model that emerged. We had very good areas under the curve. The key point I want to make here is we did a in-person, gold standard, blinded, expert psychiatrist structured SCID interview to test how well does this do against the 2 or 3 hour interview that we're used to. The answer was the positive predictive values were very good, both for the natural language processing and a couple of the structured definitions, and also 100% of the controls were controls on the SCID. And then in a follow-up genomic analysis, validated that the heritability of this was very comparable or the same as what we've seen in the PGC, and the genetic correlations with PGC bipolar disorder were very high. So that gives us some confidence. We also found that you can extract secondary phenotypes of interest, and again, have very high positive predictive values against a structured diagnostic interview. So to summarize, approach with eyes open, the fact that while there are these advantages in terms of scale and scope, it can be tricky and requires a lot of thought to extract valid phenotypes. There are real world phenotypes, but there are ascertainment biases, which we could go on about, I'm sure. We have longitudinal course and treatment outcomes, but it's also important that many healthcare systems may not be capturing all of the health events of a patient if they can go elsewhere, for example, for some of their care. We do have the possibility of phenome-wide analyses, but still high dimensionality creates multiple testing and to some degree data processing challenges. So I want to thank again all of the sites involved in PsycheMERGE. We now have EHR data for more than 12 million patients across sites, genomic data for 1 million, and what you see on the right are just some of the work groups now in progress that are tackling different aspects of the questions that we can approach in this setting. And it's again been a really vibrant, wonderful community, and we are doing all kinds of cross-site validations that are a great opportunity to interact. Here are just some of the folks who have been involved, and I know that I'm probably running over, so I'm going to turn it over to Julia so that hopefully we have time for questions as well after the next talk. So thanks so much.
Okay. All right, thanks Lea and Jordan. So like Lea said before, I am a third-year graduate student with Lea here at Vanderbilt, and I'm excited to talk to y'all today about some work that I've been doing on developing a LabWAS method and then using the LabWAS in terms of depression. So as we were discussing before, the EHR contains a tremendous amount of information on patients, including diagnoses, medications, images, vitals, and lab traits. Most of the time, whenever EHR data is used, it's in the form of the diagnoses, such as the ICD codes and fee codes that Jordan was describing. But we actually wanted to try to use the lab traits in order to get at the biology of complex traits. And we think that these lab traits and EHRs are well positioned to answer our questions about biology for a number of reasons. So first, lab results represent measured biology. So for a lot of labs, we understand their biology, and an association can help pinpoint an area to investigate further. Second, using EHRs, we can interrogate the full spectrum of clinical labs rather than subsetting to just a handful of labs. And along that same line, this can enable a hypothesis generating screen that I'll talk about later. And since we're using EHRs, we can also integrate other relevant information. We can also integrate other relevant clinical information such as medications and diagnoses. So, but working with these EHR labs does not come without challenges. So for example, a number of labs will have biologically implausible or impossible values. The same lab can have non-matching units between observations, and the same individual can have multiple observations. So then you have to decide which observation you would like to analyze. So to overcome these challenges, we developed a workflow called Quality Lab to clean EHR-derived labs for different end uses. So over here on the right is just a schematic of all the decision points of Quality Lab. So for example, you can impose an outlier filter, you can subset based on age, you can subset based on sex or race, and then you can decide which observation per individual you would like to use. We've typically used median. And then you can adjust by age for those labs and then perform a normalization. So when we apply Quality Lab in BioView, we're able to reduce the number of labs from about 6,400 to 278 labs. So with those clean labs, we're able to take them and pair them with genetics. So in this case, a polygenic score to generate a lab-wide association scan or a LabWAS. So LabWAS associates the polygenic score with each of the clean labs to find the association. So now that we have this Quality Lab and LabWAS pipeline, we wanted to validate that it was working how we expected. And to do that, we first looked at lipids. So we generated polygenic scores for 3 lipids, LDL, HDL, and triglycerides, and LabWAS each of the polygenic scores. So when we do that for LDL, so here's a LabWAS Manhattan plot. On the bottom is the grouping of the lab, and then on the y-axis is the negative log 10 p-value. So each of these triangles represents a different lab test that's being associated with the polygenic score. If the triangle is up, that means that the effect estimate was positive, and if the triangle is down, that means the effect estimate was negative. So the LDL polygenic score most strongly associates with measured LDL in our sample. When we look at the HDL polygenic score, it most strongly associates with HDL. And then finally with triglycerides, our top association is with measured triglycerides. So this was encouraging to us that we were getting out what we were putting in. But next we wanted to try looking at a complex disease trait. So we chose to look at coronary artery disease, which has a number of known risk factors. So we built the polygenic score and LabWAS the polygenic score. And when we do that, we see a number of associations that we would expect, including decreased HDL, increased glucose, and increased triglycerides. But one lab is missing, and that's LDL. So we thought that this is a statin effect. So since we're using EHRs, we were able to subset the LDL measurements to before the first dimension of a cholesterol lowering medication, and then rerun the LabWAS. And when we do that, sure enough, we see an association with pre-med LDL. So this was also encouraging to us that we were able to replicate not only the lipids, but also known risk factors for a complex trait. So next we wanted to move on to not just replication, but discovery of different biomarkers for depression. So we built a depression polygenic score and LabWAS that polygenic score, and we see a number of associations, including a couple of immune markers with white blood cell count and absolute monocyte count, as well as triglycerides, decreased HDL, increased calcium, partial thermoplastin time, and red blood cell distribution width. So this is very interesting, but a number of these traits have already been associated with depression status in the past. So we next wanted to figure out if these associations were remained after controlling for a depression diagnosis. So next we covaried the LabWAS for depression and anxiety diagnoses using Phi codes. And when we do that, we retain the associations with white blood cell count and partial thermoplastin time. So we were particularly interested in this association with white blood cell count since it was our top association, and there is a long history of depression in the immune system. So next what we wanted to do was replicate this association across the PsycheMERGE network. So here on this map that Lea showed, I have starred the sites that are participating in this project. So at Mount Sinai, Arden is running the replication. At Partners, Heather is running the replication, and at Million Veterans Program, Yorgos and Sanan are participating. So the 3 sites replicated 3 different analyses. First was the original association between depression polygenic score and white blood cell count. Next was the association covariate for depression diagnosis. And then finally, they covariated for depression and anxiety diagnoses. So as you can see, all of the meta-analyses here in black remain significant across the different tests. And interestingly, the effect estimates remain pretty steady across all of the tests and all of the sites. So here in purple is Mount Sinai. Although the association at Mount Sinai wasn't significant, they did have the smallest sample size at about 800 individuals, but still their effect estimate was really on par with the rest of the sites, so that was pretty encouraging. And this was really fun for me to see that it wasn't just a one-off association in BioView, but it really did replicate across a few different sites and in the meta-analysis. So next, we wanted to understand what potential pathways were working between white blood cell count and depression. So we came up with 3 different pathways and hypotheses and a way to test those hypotheses. So the first pathway could be that depression genetics are affecting white blood cell count biology. And so we tested that by repeating our original association, looking at the depression polygenic score and white blood cell count. But next, it could be that white blood cell count genetics are driving the association because they affect depression diagnosis. So then we built a polygenic score for white blood cell count and associated it with depression diagnosis. Or finally, it could be that white blood cell count genetic elements within the depression polygenic score are driving the association. So in order to remove those white blood cell count genetic elements, we conditioned the depression summary statistics on white blood cell count summary statistics and rebuilt a conditional polygenic score and associated that conditional polygenic score with measured white blood cell count. So the results from the direction analysis looks like this. So here at the top is our original association between depression polygenic score and white blood cell count with MVP, BioView, and Partners, and then in black our meta-analysis. So across our 3 sites, it remains significant. And then the association between white blood cell count, polygenic score, and depression diagnosis. So at 2 different sites, BioView and Partners, it was not significantly associated, but at MVP in our meta-analysis, it was significantly associated. However, this effect estimate is less than half of what we saw in the original association. And then finally, whenever we condition the depression summary statistics on white blood cell count genetics and redo the association with the conditional polygenic score, it remains associated with white blood cell count with a very similar effect estimate like what we were seeing in the original association. So this is kind of, my takeaway from this is that it's really the depression genetics that's driving the association with white blood cell count rather than white blood cell count genetics driving the association. Okay, so hopefully I left enough time for a discussion and some questions. So to wrap up, here's some conclusions from my part. So Quality Lab and LabWAS can be used to recapitulate known clinical biomarkers and also find new biomarker associations. The depression polygenic score associated with an increase in white blood cell count even after controlling for depression and anxiety diagnoses across sites. And I also want to mention that we also controlled for a number of common phenotypes between depression and white blood cell count, and the association did remain. And then finally, it looks like depression genetics drives the association with white blood cell count rather than white blood cell count genetics driving the association. So with that, I just want to thank Lea and Jordan for their awesome work leading the PsycheMERGE network, and also all of the collaborators that have replicated the analyses, Yorgos, Sana, Heather, and Arden. So, thank you.
Wonderful. Thank you so much, Julia and Jordan. So we have about 6 minutes for questions. I know Jordan has to go, has a hard stop at 11. So maybe we'll make sure to get questions for Jordan in first, and it looks like everybody has been unmuted, so go right ahead and speak up if you have a question.
Can I ask about, oh, sorry, go ahead.
It's a free-for-all. Okay. Maybe I can call out names because it looks like people are raising their hands. So Henry, do you want to, I think I heard your voice first. Do you want
I just want to ask, you looked at WBCs. Are you able to fractionate those in terms of lymphocytes and PMNs? Because I think that would be informative.
Yeah, we are able to look at the differentials, and that's something that we're looking at right now. So in the original lab WAS, the monocyte count was associated with the depression polygenic score, but we're also able to look at lymphocytes and neutrophils that also seem to be playing a role in the relationship between the polygenic score and white blood cell count.
Thank you.
All right, Talia?
Hi, thanks guys, great talks, really fascinating work. So I have one for Jordan about the PHIWAS, and apologies, this might be a really stupid question, but I was just quite intrigued by the fact that the schizophrenia PRS seemed to be more strongly associated with anxiety and depression than with schizophrenia itself. And I wanted your take on whether that's real or whether it's something to do with the fact that these disorders are so much more common, or a combination of the two. It's not an analysis I'm that familiar with, so I'd appreciate it.
Yeah, no, thanks. And I realized I tried to answer your question in the chat, but it only went to the panelists. So yes, I think the I think that the answer is what you just said, which is that it was really a function of the sample size. So we had many more anxiety, depression cases, and if you look at the odds ratios or the effect sizes, it's definitely higher for schizophrenia or psychosis. So that effect is stronger, but by virtue of sample size, the p-values are much smaller.
Okay, great, thank you. So if we look at the associations by effect size rather than by significance, then it comes out as you'd expect.
Exactly.
That's right. And in fact, it was the strongest odds ratio in the entire PHIWAS was with schizophrenia.
Schizophrenia was, right. Yeah. Okay, lovely. And I missed it, apologies. Is that published, that paper?
Mm-hmm.
That one is, yep. It's American Journal from late last year.
Okay, I'll look for that then. Lovely, thank you. I've got to go, but really enjoyed your talks. Thanks very much.
Thanks.
Talia.
Bye.
I see there's a question in the Q&A, actually. How do you remove privacy information before using EHR data? And did the EHR from different sites have the same data structure? Good question. The model for PsycheMERGE is a federated analysis model. So the data do not leave each site. What we have done is go to great lengths to develop common data pipelines and standard operating procedures for the analysis, so that we do lots of cross-checking about the validity of cross, but we avoid that problem of actually transmitting individual level data. The EHR from the different sites data structure, essentially the answer is yes, and there are formal ways to do this through what are called common data models, which our sites use. Familiar ones would be OMOP and I2B2 that allow you to essentially interoperate the different data structures.
There was another question to Julia asking about the variance explained by the polygenic score in terms of white blood cell count. So Julia, do you want to just mention a little bit about that?
Yeah, so the variance explained by the polygenic score for depression is pretty low. If I remember right, it's somewhere between 1% to 2% of the variance that we're explaining. And then when you're looking at the variance explained of white blood cell count by the depression polygenic score, I think it's less than 1%. So it's not a lot of variance that explained, but it still remains significant.
There's another question. Julia, have you tried Mendelian randomization techniques with respect to white blood cell count and depression?
Yeah, so a while ago I did try summary level Mendelian randomization between depression and white blood cell count, and there was not a significant association in either direction. I haven't tried like I guess since we're using measured white blood cell count, we could use like a single study, a single sample Mendelian randomization, but I haven't tried that.
All right. And then there's another one, white blood cell count conditional. Do you expect this one to be as significant as the white blood cell count PGS PRS? So I think this would be for the depression PRS, as it's roughly.
I was asking there. So if when you are doing you are regressing WBC on the PGS given WBC PRS, yeah, you should obtain roughly the same statistical significance as doing WBC on the PGS PRS, because it's roughly a scale transformation of the other. And I think that's what you obtain there, yeah?
Yeah, so sorry, I'm not quite sure what you mean by PGS PRS.
If Julia is going back to the, can you go back to your slides?
Sure.
Yeah, where you tried the three models?
Yeah.
I just scribbled them down and.
Oh, sorry.
Okay, this, yeah, so okay, depression PGS, okay, so that's right. So the first and the third one,
Gotcha.
they should be roughly similar in a way, because when you are doing depression PGS that given WBC, yeah, that is a scale transformation of WBC.
So I guess maybe this isn't clear, but we actually conditioned the depression summary statistics on white blood cell count summary statistics. It's not conditioned on the measured lab trait. It's conditioned on the genetic.
On the statistics. Yeah, but yeah. That's what I'm saying. That probably this captures roughly the same as the first one.
Yeah, it looks that it is the same. Yeah, but yeah.
Yeah, I mean, the main goal of that was really to make sure that what we were seeing in the depression polygenic score was not being driven simply by, variants that are associated with depression that are also happen to be in like hematopoietic pathways that would be known regulators of white blood cell count already.
Okay, so probably for that one, you have to subtract the conditionals from them, but yeah, I think that, okay, yeah, thank you.
Sure. Okay, and then it looks like our last question is from James Walters. What proportion of patients do you have DNA for, and have you looked to see how these people are similar or different to those without DNA? So that's a very good question, and it's different across different sites. I can tell you that for Vanderbilt, the way that we do the DNA collection is that it's obtained from leftover blood from a clinical blood draw. So in our outpatient clinics, if anybody who comes into our outpatient clinics has the opportunity to enroll in BioView, which means that when they have lab work done, they agree that the remaining blood can be banked as opposed to discarded. And so that influences our ascertainment for collection in a couple of ways. One is that we do tend to have fewer children, for example, so because kids get fewer blood draws, and when they get blood draws, they usually have less blood drawn. So there's usually less opportunity for them to be involved in the biobank, although we do We do have about 25,000 kids in the biobank. The other thing is that people who come in just for inpatient procedures are typically not given the enrollment into the biobank. So it's really primarily our outpatient patients who have the opportunity to enroll in the biobank, but then if they have inpatient procedures, of course, that is also going to be reflected in their electronic health record. So we'll still have that data. So in terms of the proportion of patients, we have about 3 million individuals in our EHR, about 280,000 in with DNA extracted, and then out of those, about 100,000 with genotype data available. So those are the Vanderbilt numbers, but they tend to be pretty similar across other biobanks that have been established as long as Vanderbilt has. So I think it's now 10:06. So I really appreciate everybody staying on for a few more minutes and engaging in discussion. This was a wonderful set of presentations, and I am very grateful to the PGC for hosting it. So Tammy, I think, do we leave it to you to close the meeting?
Yes, ma'am.
Okay. All right. Thank you, everybody.
Mm-hmm.